What I'd like to do with this video is not so much go over any details about the skeleton, but talk about something you should be aware of in the laboratory, or even if you're at home studying anatomy using figures. And that is that you're going to find a lot of variability in the images that you look at and in the specimens you look at. So I'm going to show you some examples of what I'm talking about, using a plastic skull, using a figure, using a model that illustrates the position of blood vessels, and finally using an actual skull that's been disarticulated and set up. So to start off with, here we have an example of a specimen, which is a kind of idealized specimen because it is a plastic model of an original human skull. And it's done pretty well, but it's based on an individual. And there is, from person to person, a lot of individual variation. So you just look at a face, and which is easily recognizable unless it's an identical twin. Faces will vary in many ways, some subtle, some not so subtle. And that's an example of the variation that you find in all of biology. And you're certainly going to see this variation reflected also in the skull. So if you're looking at one skull and it's from one individual, it may be very different in many ways from the skull of another individual. So that's one thing, the individual variation. So here's another example. It's a drawing, obviously. It comes from Gray's Anatomy, the 1918 edition. And we're going to see that if we put this side by side the plastic skull, we're going to see differences. So here is the skull next to the Gray's figure. And if you look at each, you can see there are differences. There are notable differences in between the two. So if you were to see these two individuals in real life, you would be able to tell one from the other pretty readily. But there are some other subtle differences that gets me to the major point I want to make. There are certain features that you're going to find that are going to look very different as you go from one specimen to the next. So let's look at a couple of examples. So if we zoom in to the Gray's figure, you'll notice that there is a feature that we find above the orbit, which is referred to as a supraorbital foramen. And if you look at the left eye, above the left orbit, you'll see it is indeed a foramen. However, when you go over to the right eye, you don't see it. You don't see a foramen. What you see is a notch. So if you were to describe it, you wouldn't call it a foramen since it isn't an opening in the bone, but it's a notch. And so here we can see that you can actually see this variation in an individual skull, one side different than the other, the right being different than the left. If we go over to the plastic skull and look at it, notice that you don't have a supraorbital foramen. What you have are two supraorbital notches. So this illustrates how this variation can occur. And if you slavishly study and assume that a foramen is a foramen, a notch is a notch, and you're looking at a figure or a skull, you're going to be confused. So does it matter if it's a supraorbital notch or a supraorbital foramen? Well, if we take a look at this skull, this model, it's meant to illustrate the vasculature of the skull. And if we zoom in on the place where we have our supraorbital foramen or notch, we see in this instance we have a foramen, the blood vessels going through an opening, which would be a foramen. However, you can just as easily imagine there being a notch there and that same blood vessel just going around the bend of the notch. So whether it's a foramen or a notch, it serves a purpose, and the purpose is served by either or. Let's take a look at another example. If we look at these two skulls again, notice that the frontal bone, the bone of the forehead, in both the figure and in the plastic skull is just one bone. There is no separation by a suture, in other words, it's just one bone. We'll discover later that the frontal bone comes about as the result of the fusion of two bones as they develop in the embryo and the fetus. However, if we take a look at the actual human skull bones that are part of this articulated or disarticulated skull, notice that the frontal bone has a suture that goes right down the center, separating it into two bones. This suture is referred to as the Metabolic Suture. It's also known as the frontal suture. And it is uncommon to see this in a mature skull. So what is this? Well, is this an abnormality? Well, not necessarily. It probably didn't cause the individual any problems in life. However, it is something that is unusual and different as you look at different skulls. So this would be considered an anomaly, which does not necessarily have any pathological or serious consequences for the individual. But this is something that you'll see and occasionally, and if you didn't know better, you looked at this skull and you thought it was an example of all skulls, you would think that all skulls should have this suture, but you would find that that isn't the case at all.
So let me summarize the key point of this video, and that is that you need to look at as many different specimens, as many different individuals as possible if you really want to understand anatomy, because of the presence of this variability. And the sources of the variability include a bilateral asymmetry, which means that your right side is different than your left side. This is obvious, for example, when you look at your right hand and compare it to your left. They're not identical. One is a mirror image of the other. And if you are looking at the bones from a left hand and you were referring to a textbook that had a picture showing the right hand, you might be very confused trying to match the bones that, for example, you find in the wrist if you didn't realize that one was a mirror image of the other. Another thing is that you're going to see individual variation. If you have bones from actual human specimens, there's always going to be a lot of variation. And even if you look at these plastic models, if the plastic models come from different individuals, you're going to see variation. So you should appreciate that variation and understand it. Sexual dimorphism is another thing. The male is different than the female in many characteristics. Obviously, with the reproductive organs, you're going to see very different organs there. But even with the skull, for example, the skull of the male is different than the skull of the female, and you can sometimes tell if an individual was a male or female just looking at the skull, because of the fact that the male skull tends to have more distinct, robust features compared to the female, where the features tend to be more delicate. Another factor is development. Young bones are different than old bones. For example, in young bones, you'll find growth plates in the long bones, which you're not going to find in the older bones, because these growth plates are going to disappear. You also are going to have to deal with the possibility that a specimen, an actual specimen that you're looking at, may have had pathology going on. So, for example, older bones may show signs of osteoporosis that leads to them being more fragile, more easily broken. And you may look at these older bones and think that an opening is a real opening when it's actually due to the weakening of the bone that caused a piece to fall out. Then finally, you might see some trauma where, a broken bone that didn't heal properly, and that would, if you looked at that specimen and didn't understand the trauma that occurred and how the bones didn't heal properly, you might be led to think that something that shouldn't be there is there, that the abnormality due to the trauma is real. So just, again, the central point is you need to keep in mind this variation exists and look at as many things as you can.